So next presentation is going to be given by Hao Chi from the Chinese Academy of Sciences. That's one of the proceedings presentations, pNovo 3 precise de novo peptide sequencing using a learning to rank framework.
Okay, thank you for the introduction. Morning, everyone. I'm happy to introduce our new study about the precise de novo sequencing, pNovo 3. First, I want to briefly introduce peptide and protein identification using the tandem mass spectrometry. Given one protein sample, we want to know how many proteins in this sample and what are their sequences. First of all, we digest proteins to peptides using some enzymes, for example, trypsin. Then, mass spectrometer is used to generate tandem mass spectra. Next, we can use computational approaches, analyze this data, and identify peptides. Finally, we can know the protein sequences by assembling the peptides together or use some protein inference algorithms. In the whole workflow, the key step is how we can interpret the tandem mass spectra to peptides. If we have a sequence database or a spectral library, we can search the spectra against these databases, compare each spectrum with some theoretical peptide candidates, and choose the best scored peptides. This is called database search, the dominant approach in the routine MS data analysis. De novo sequencing is an alternative approach which identifies peptide sequences directly from the mass spectra without any databases. A general method for de novo sequencing is constructing a spectrum graph for each given spectrum. For example, given one peptide and the corresponding spectrum, the B and Y ion series are in the spectrum so that we can see the mass difference between two adjacent B and Y ions equals to the corresponding amino acid residues. So the de novo sequencing problem can be transformed into the finding the best path in the spectrum graph, where each node is generated based on the peaks. And if the mass difference between two nodes equals to some amino acid residues, we can connect them by an edge. If we can find the best path given the optimization criterion, the corresponding peptide can be restored simply as the answer of the spectrum. We have developed a de novo sequencing algorithm of HCD spectra called pNovo in 2010, and further developed a few other related algorithms and tools to make the performance of de novo sequencing better. For example, we combined the HCD and the ETD spectra, or the spectra produced by the two mirror proteases, trypsin and lyserginase, to increase the spectrum quality. In addition, we designed tools to open de novo sequencing, Open pNovo, and the precision evaluation tool, pSite. Although we have developed these tools to improve the performance of pNovo, one problem has not been solved well yet. That is the missing of some key fragment ions. For example, given such a peptide spectrum match, we can see that no fragment ions between P and E were detected, B9 and Y3. So the best thing that de novo sequencing can do is to report two peptide sequences different from PE and EP with equal credibility. Actually, as the database of this sample is available, so we can know the first one is correct because it can be matched exactly with the database. But such information cannot be directly used by de novo sequencing. So if we want to distinguish between these two peptide sequences, we must find some key features from the sequences themselves. Dr. Zubrov investigated the performance of peptide fragmentation in CAD data, and from this figure, we can see that the fragment ions at the N-terminal of the proteins are easily observed and with high intensities. However, the C-terminal, the intensities of the C-terminal fragment ions of the proteins are very low. They are quite different. So, based on this clue, we can simply speculate that PE should be more probable to be the correct one without any database information, because the mixed fragment ions between P and E are indeed not easily to detect in the real MS data. Therefore, we can take advantage of more fragmentation patterns learned from the MS data. Instead of learning some simple rules from the statistical result, a lot of deep learning-based algorithms are proposed recently to generate more precise fragmentation patterns and predict theoretical spectra. The algorithm proposed by our group is called pDeep, which is used in pNovo, pNovo 3. And the latest improvement, pDeep 2, which uses the transfer learning approach to predict the theoretical spectra with modifications based on the prediction of unmodified peptides. It has just published online. Then we can use pDeep to predict the theoretical spectra of two peptides. For the correct one, we can see that the two PSMs looks very similar, the real spectrum and the predict spectrum. More importantly, pDeep predicted a very low Y3 ion and no B9 ions. They are quite consistent with the real spectrum. Lowever, for the incorrect one, we can see that pDeep predict a very high Y3 ion because it really located at the N-terminal of proteins. The B9 ion is also predicted with a medium intensity. So we can conclude that the similarity between these two PSMs is much lower than the two PSMs shown in the last slide. So, based on the use of pDeep, we developed the de novo sequencing tool pNovo 3. Use the traditional workflow of pNovo, we can get a few top-ranked peptide candidates for each spectrum. Then, we use pDeep to predict the theoretical spectra for these peptides and calculate the similarities between the real spectrum and each of the predicted spectra. 3 similarities are calculated, cosine, Pearson, and Spearman. In addition, we added 2 gap-related features, which indicate the frequency of missing fragment ions between 2 amino acids, and they can be calculated from the statistical results of well-annotated data. With these features, a model can be trained by SVM rank, and we can get a new order of the peptide candidates for each spectrum. At last, we use a spectral merging approach to further update the top 1 results for some spectra, which will be introduced later. So how can we get the gap features, uppercase G1 and G2? First, we can compute the probabilities of losing fragment ions between 2 amino acids based on the statistics. G1, the lowercase G1, given 2 adjacent amino acids, A1, A2, the lowercase G1 is the number of A1, A2 between which the ions are missing, divided by the total number of A1, A2 in the dataset. The lowercase G2 same as G1, but only for the N-terminal 2 amino acids of the peptides, because the order of the N-terminal 2 amino acid is more error-prone. So we can calculate the uppercase G1 and G2. G1 is the arithmetic mean value of all gaps G1 found in the PSM, and the uppercase G2 is simply the value of the lowercase G2. These 2 features are used together with the similarity values for the following SVM rank model. Okay, here is an example of the feature values. For the 2 PSMs mentioned earlier, we can extract the 5 features. For the correct peptide, the 3 similarities are all greater than 0.97, which are much higher than those of the incorrect ones. The G1 feature of the correct peptide is also greater than the incorrect one, suggesting that PE is more likely to produce a gap compared with EP. Two G2 values are both the default value 1.0 because no gaps were found at the N-terminal of both peptides. As a final procedure of pNovo 3, we use a spectral merging approach to further increase the performance. For example, given one PSM, the spectrum S1 and the peptide ADCEFK, in the real MS dataset, we may find 5 other different spectra with the same precursor mass. If one of these 5 peptides can be well matched with S1, this peptide can also be considered as one candidate of S1, but it does not be found by the de novo sequencing. So we can find that in this spectrum group containing 6 spectra, there were 3 different peptide candidates which have different number of supported spectra and different mean score of the original score. Then another SVM rank model is used to rank these peptides, and finally the top 1 peptide is considered as the final answer of S1. In this study, we use 7 high-resolution MS datasets for the performance evaluation. All of them can be downloaded from the proteomics change repository. The first dataset is used for training the SVM rank model, while the other 6 datasets are used for the performance evaluation as test datasets. For each dataset, we use pECFun and pECCB to search them and extract the spectra that consistently identified by these 2 engines as the benchmark data. First, we compare pNovo with 3 other de novo sequencing tools. Compare pNovo 3 with pNovo, pX, and novo. Actually, the top 10 peptide candidates between pNovo and pNovo 3 are the same, and pNovo 3 adjusts their ranks to each spectrum. As shown in this table, about 60% of spectra in the benchmark datasets can be correctly sequenced by pNovo 3, which is significantly higher than those of the other 3 algorithms. Further, we have checked the recall varied from top 1 to top 10, as novo only reported the top 1 peptide sequence for each spectrum. We only compare pNovo 3 with pNovo and pX. Compared pNovo and pX, we can see that although the recall of top 10 results of pNovo was slightly higher than pX in most cases, the top 1 result of pNovo is similar or even a little worse than pX. It shows that the original scoring method in pNovo was less effective to distinguish between the similar peptide candidates. That's why we developed pNovo 3. As a result, the refined scoring method in pNovo 3 seems much more powerful and yielded a large difference of recall compared with pNovo and pX, especially for the top 1 results, which are more important for the real biological discoveries. Finally, we compared pNovo 3 with the latest de novo sequencing algorithm using deep learning, DeepNovo. As DeepNovo uses a different model for the 9 high-resolution test datasets in its paper, it trained 9 different models were trained for each of the 9 datasets. So we just downloaded the original results of pNovo rather than reanalysis the dataset using the unified model provided in the software. Then we tested pNovo 3 with the same 9 datasets to make a fair comparison. The benchmark strategy was the same as that shown before. As shown in this table, on the new benchmark datasets, pNovo perform better than PIX and novo. It shows the effect of the deep learning approach. At the same time, pNovo 3 recorded about 13% more spectra of the total spectra. So the gap between pNovo 3 and pNovo, we think, mainly due to the different way we use deep learning. pNovo combines the deep learning approach and the dynamic programming together. It predicts amino acid one by one. But pNovo splits the whole workflow into 2 steps. First, it uses the traditional de novo sequencing algorithm spectrum graph approach to generate a few top-ranked peptide candidates. And then it uses a deep learning approach, for example, pDeep, to extract more features to refine the scoring function. As the traditional de novo sequencing, the spectrum graph approach, it has been widely used in the past decades, and it may be more mature. So we use such 2-step workflow. First, we use the pNovo, and then we extract more features to train a more powerful scoring function in pNovo 3. So that's the main content of our work, and let me make a brief summary. Based on the existing workflow of pNovo, we developed pNovo 3. We built a learning-to-rank model to re-rank the original results of de novo sequencing, use the deep learning approach, pDeep, to extract new features. And finally, we proposed a spectrum merging method to further improve the performance of pNovo 3. Compared with a few other de novo sequencing tools, pNovo 3 records significantly more spectra on several high-resolution MS datasets. Finally, I want to thank all p-funders in our group. We have also developed a few other tools in the pFund Studio, including the common database search engine pFund, the cross-linking data search engine plink, and the N-glyco proteomics data search engine pGlyco. And we welcome you and your colleagues to try our tools. Thank you very much for your attention.
So have you separately quantified how well your tool works if you turn off the spectrum merging? In other words, how much of your improvement is due to the deep learning versus the spectrum merging?
Yeah. I have tested because there are 2 main points. One is use the deep learning-based features, and one is spectrum merging. Yes. We use, if we turn off the deep learning or the merging, spectrum merging, about I remember about about half spectra cannot be recorded. So both of the two points are very useful in this framework. Yeah.
Yes, very nice, thank you. What are the type of errors you have? Like, if you look at what you're not correctly sequencing, do you have any intuition of what are the type of problems you're facing now in the remaining 50% or 40%?
I'm sorry. 40%? Yes.
So this is fantastic, yeah. And now you're reaching about 50%.
Yeah, thank
Drop.
you.
Have you looked in the remaining 50% to see what are the type of errors? These are swaps, or do you have like almost all amino acids and they're just 2 swaps, or is it completely different? Or is it due to, I don't know, modification of the peptides? This is the question.
Oh, yeah. This is always the hardest problem in de novo sequencing because the benchmark is from database search. Database search is more powerful and it can interpret some medium or low quality of spectra. So I think maybe the other half of spectra, it may be the low quality spectra maybe. So they can be interpreted by database search, but not de novo sequencing. Yeah.
Unfortunately, we have to move on. We are running already 5 minutes late. Thank you so much.
Thank you.